In this video, we're heading out to Puget Sound on the small research vessel RV Noctiluca. Safety first, these full-body Mustang suits float in case they get knocked overboard by a wave or a giant octopus. Brian starts up the boat and we leave Friday Harbor behind and head over towards our first sampling station on East Sound. Today, we'll sample 10 stations, deploying a CTD at each one. The CTD, shown here, measures conductivity, temperature, and depth. Conductivity is a way of measuring salinity, or the saltiness of the water. Temperature, well, is the temperature of the water. And depth shows the changes in these two parameters as the CTD goes deeper into the water. These data are then downloaded and analyzed. The top of the graph represents the surface of the water, and the bottom of the lines represent how deep the CTD went. The red lines on the left represent salinity. As you can see, it's a straight line, meaning the salt content of the water is consistent throughout the water column. The green lines represent temperature, and this graph shows the water getting colder with depth. The black lines are from a fluorometer connected to the CTD and represent chlorophyll, which essentially tells us how many phytoplankton are in the water at a given depth. Now, the term water column is just as it sounds. It's a column of water that the CTD goes down through from the surface, much like if you were to stick a knife or your finger straight down into a layered cake. You would be able to tell where the icing stopped and the cake started and so on, whereas the CTD tells us the structure of the water column, the amount of light and nutrients, and where the phytoplankton are. Since we want to collect phytoplankton, we find where chlorophyll is most abundant in the water column. Then we deploy these gray Niskin bottles to that depth and collect water to bring back to the lab with us to use for experiments.